Yes, photosynthesis in higher plants we have started in the previous class itself. Beginning, let's recap everything once, okay, in 10 minutes. First, about discoveries beta. We have learned the entire discoveries by taking the standard equation of photosynthesis. Sit straight everyone. Carbon dioxide is essential for the process of photosynthesis set by Moll's half-leaf experiment. Carbon dioxide and sunlight are essential for photosynthesis by Jan Ingenhousz. Oxygen evolved from the process of photosynthesis by Jan Ingenhousz itself. Oxygen evolved from the process of photosynthesis by Jan Ingenhousz. By what experiment? Hydrilla leaf experiment or inverted funnel experiment. Okay. And then oxygen discovery by Joseph Priestley. By what experiment? Bell jar experiment. Who discovered that glucose is the primary product of photosynthesis? Von Sachs. And who said that the oxygen is being evolved is coming from water and not from carbon dioxide? It is C.B. Van Til. C.B. Van Til has conducted what experiment? Bacterial photosynthesis. What is the electron donor of bacterial photosynthesis? H2S. Electron donor for oxygenic photosynthesis is H2O. Clear with the basic discoveries? So, it is Jan Ingenhousz who said that sunlight is essential. From that, it is E.W. Engelmann who has derived that in two regions, more amount of sunlight is being captured. Okay, that is in what regions? Blue region and red region of sunlight. By using what model organism? Look here everyone. By using what model organism? Cladophora. For every experiment there will be one model organism. What is the model organism for inverted leaf experiment? Hydrilla. Model organism for this experiment? Cladophora. And aerobic bacteria as well. More amount of aerobic bacteria got accumulated in what regions? Blue and red regions. Hence it is proved that in those two regions maximum amount of oxygen is evolved, maximum amount of photosynthesis happened. Thereby he has derived out what? Action spectra and absorption spectra. To know about action spectra and absorption spectra, we have learnt about pigments. We have two main types of pigments. One is fat soluble, second is water soluble. Water soluble pigments include? Two types of them, what are they? Phycoerythrin, phycocyanin. Fat soluble pigments include chlorophyll, right ma? And then anthocyanins as well you can take. Now, action spectra and absorption spectra. Amongst these three graphs, which is showing absorption spectra? First graph. What information you will get from the absorption spectra? Different pigments absorbing light at different wavelengths. On x-axis what is plotted out? Wavelength of sunlight. On y-axis absorbance of chlorophyll pigments in absorption spectra. In action spectra, amount of oxygen that is evolved. What is plotted on x-axis, what is plotted on y-axis of absorption spectra and action spectra is very important. Okay. So, here what are the two major peaks that you observe for which pigment? Two major peaks. Chlorophyll A at two different wavelengths. Chlorophyll B will show one major peak and one minor peak. Carotenoids show two major peaks in between 400 to 500 nanometers. Remember that carefully, Nana. At x-axis, y-axis, what is that being plot? Okay. And what are the major peaks shown? Because this graph will be given you directly and they will be asking you for the plotting of different pigments. You have to know. Okay. And then about action spectra. Action spectra will give you the information only about that one pigment which is conducting maximum amount of photosynthesis. It is chlorophyll A. Okay. Two major peaks, one in between 400 to 500 and one in between 600 to 700 nanometers. And then in the last graph you can see chlorophyll A is the pigment which is coinciding both with the absorption spectra and action spectra. Clear with this? Any doubts anywhere ma? And then we have clearly dealt with the chloroplast organelle which is double membrane bound. The space in between two membranes is known as periplastirial space. Inside the chloroplast you have fluid filled matrix. That fluid filled matrix is addressed as stroma. It consists of starch granules, lipid granules, RNA and proteins. These are the molecules that are present inside the stroma. Presence of 70S ribosomes, double-stranded circular DNA shows that these organelles have evolved from prokaryotes, which is clearly explained in endosymbiotic theory. This organelle is semi-autonomous cell organelle or self-replicating cell organelle. And membranous sac-like structures are thylakoids. Thylakoid membrane is also known as thylakoid lamellae, which consists of light harvesting complexes known as photosystems. Stalk of this sac-like structure is grana. One grana adjoined to the other grana by stroma lamellae. Now we are clear with the organelle, right? Now we will move on to the light harvesting complexes. In thylakoid lamellae or thylakoid membrane, the light harvesting complexes which are present are known as photosystem 2 and photosystem 1. Here if you see, in any of the light harvesting complexes, you have two types of pigments. One is known as core pigments, second set of pigments are accessory or antenna pigments. Accessory or antenna pigments include chlorophyll B, carotenoids and xanthophylls. Core pigment includes and there where the reaction center is present, okay, where all these different accessory or antenna pigments will gain the energy at different wavelengths, lower wavelengths, 400 to 500 nanometers. They will take the photons energy and they will get excited, give it to the reaction center, that is to the core pigment, that is chlorophyll A. Clear, right? This is the story of two photosystems which are present in the thylakoid membrane. Now about different reactions that happen in the photosynthesis, very important. Light reactions are also known as light dependent reactions, and this phase is also addressed as photochemical phase. Any doubt? She have to move a bit, clear, okay, yeah. Photochemical phase. Why is it known as photochemical phase? Because physical energy is converted to chemical energy. Physical energy in the form of photons is converted to chemical energy, that is ATP and NADPH. Light dependent reactions include three major reactions. First is absorption of light in the form of photons. Where does this happen? Inside grana, inside thylakoid membrane. Second, photolysis of water, inner membrane of thylakoid towards the lumen. Formation of ATP and NADPH in stroma. Where does light reactions takes place? Whole and sole in grana of the chloroplast. Next, biosynthetic pathway. What is biosynthetic phase? Synthesizing the glucose molecule or glucose formation. Here what exactly happens is carbon reduction or carbon dioxide assimilation. Be here all, be here. Carbon reduction or carbon dioxide assimilation. Carbon will get reduced to CO2 will get reduced to C6H12O6. Addition of protons is what? Reduction only, right? Carbon is reducing, carbon dioxide getting fixed, carbon assimilation, one and the same. Carbon reduction, carbon assimilation, carbon dioxide fixation, one and the same. That occurs in which phase, Nana? Biosynthetic phase or light independent reactions. These are addressed as light independent reactions because they are not directly dependent on light, they are dependent on the products of light reactions. What are the products of light reactions, beta? ATP, NADPH and oxygen, okay. And here for the glucose formation there are many different cycles operated in different plants. In all the plants there is one common cycle that is operated, what is that? C3 cycle. But it is majorly present, only one C3 cycle without any other cycles are present in temperate plants. Only C3 cycle is present in temperate plants. C3 cycle is present in all the plants. Along with C3 cycle, C4 cycle is present in tropical plants. Along with C3 cycle, CAM cycle is present in desert. Remember that, okay. And then you will also learn about photorespiration, which is operated only in C3 plants. This is about biosynthetic phase. Clear everybody? I'll give you 1 minute time, just fix this in your mind. Just look at the complete flow chart, fix it in your mind. Done, right? Now, let's begin with the light reactions, okay? For that, what did we learn? Non-cyclic photophosphorylation, right? Okay, first, consider this as thylakoid membrane or thylakoid lamellae, and this is thylakoid lumen. First step is what? Absorption of light in the form of photons. It is done by light harvesting complexes which are present over here. Utilizing that physical energy, photolysis of water happens inside the lumen. Oxygen evolving complex, by oxygen evolving complex. This gives out, one water molecule gives out 2 protons, 2 electrons and an oxygen atom. 2 water molecules will give out 4 protons, 4 electrons and 1 oxygen molecule. Okay. And this photolysis of water will supplement the continuous flow of electrons. Uninterrupted flow of electrons is given by photolysis of water. That statement you remember. We may get in statement based question. Okay. And here the electrons, primary acceptor of electron is what? Pheophytin. Electrons get excited, they reach reaction center, that is chlorophyll A. From there, the primary electron acceptor is pheophytin. And then the electrons are given to proton carrier. Plastoquinone, here it is carrying protons, right? Proton carrier, that is plastoquinone. Protons are being given and this turns to plastohydroquinone. Protons are sent to lumen. Electrons are given to what, ma? Cytochrome B6F. Cytochrome B6F to again plastocyanine. This is the sequential flow of electrons you have to remember. Fine, right? What is the ultimate electron acceptor of photosystem 2? Plastocyanine is the ultimate electron acceptor of photosystem 2. From here, the electrons are being submitted to chlorophyll A, reaction center of photosystem 1. Okay, this reaction center maximum absorption of wavelength is what? 700 nanometers. Here reaction center maximum absorption is 680 nanometers. So you can also address this as P680 and this as P700. Okay, and this primary electron acceptor of PS1 is ferridoxin, then to ferrous sulfur complexes, and the ultimate electron acceptor is NADP plus. Okay, NADP plus is present towards stroma side of the thylakoid membrane. Okay, stroma side of the thylakoid membrane where NADP plus is reduced to NADPH plus H plus. By what enzyme? NADP reductase. By the enzyme NADP reductase, NADP plus is reduced to NADPH plus H plus. Clear with the first two reactions? First is absorption of light in the form of photons, second is photolysis of water, third is formation of assimilatory power. You got to know how NADPH is formed. After this, we have also learnt about formation of ATP from chemiosmotic hypothesis, which is given by Peter Mitchell. What are the prerequisites for chemiosmotic hypothesis? A membrane is required, a membrane pump is required, that is F0, F1 channel, proton gradient is required. Okay. So these are the prerequisites of what? That is ATP synthase complex of chemiosmotic hypothesis. Three major reasons you have to know, because on one side one complete page explains about this chemiosmotic hypothesis only. Okay. Listen carefully. First point, because of photolysis of water in thylakoid lumen, more amount of protons are present. Second point, protons whichever are present in the stroma are being sent into the lumen. Be here all, from stroma are sent into the lumen, okay, via what? Plastoquinone. Okay, protons are sent into the lumen via plastoquinone. Third, whichever little amount of protons are present in the stroma, they are reduced to NADPH. So, because of these three reasons, high amount of protons accumulated in the thylakoid lumen and low amount of protons you can see in the stroma, because of which what is generated with a proton gradient. F0 F1 pump is activated, which this pump is known as ATP synthase pump, or it is also known as oxysomes. Okay, ATP synthase or oxysomes or F0 F1 channel, where F0 is embedded inside the membrane, F1 is towards the stroma. In a single run, how many protons are pumped? 4 protons, which are required to the formation of 1 ATP. ADP and inorganic phosphate will make 1 ATP. Clear? This is all about chemiosmotic hypothesis. Fine, right? Now we will once look into cyclic photophosphorylation. Occurs where? In bacterial photosynthesis, in stroma lamellae. For non-cyclic photophosphorylation, it occurs in thylakoid lamellae, where two photosystems are present, PS1 and PS2. But here this occurs in stroma lamellae, only one photosystem is present, that is PS1. So, the maximum wavelength will be 700 nanometers, which is P700. Got it right? Okay. So, when the photons are being pumped and first electrons get excited, right? They will get excited to what? No photolysis of water here. There is no photolysis of water, no formation of NADPH. ATP will be formed when there is electron flow from cytochrome B6F to plastocyanin. So, from chlorophyll A to ferredoxin. So, here primary electron acceptor is ferredoxin. Ferredoxin to plastoquinone to cytochrome B6, then to plastocyanin, from plastocyanin back to chlorophyll A. So, this is a cyclic flow of electrons. Clear? Take the points regards to cyclic photophosphorylation, where is it operated, and take the sequence of energy flow. Mostly you will get the sequence of electron flow from non-cyclic only. Here you remember these major points, followed by what is the primary electron acceptor and last electron acceptor. Plastocyanin is giving back electrons to chlorophyll A. That's all. Done? Now, biosynthetic phase. Listen carefully, many a times half the set of questions, if you have to take all the previous year questions from photosynthesis of higher plants, most of the questions are from non-cyclic photophosphorylation of light reactions, okay, and half the set of questions are from biosynthetic phase. It can be from C3, C4, CAM, or from C2, or from bioenergetics of C3, C4 and CAM. So, listen cautiously. This is also known as what, ma? Biosynthetic phase. What happens in this phase, beta? Glucose formation, that is carbon dioxide fixation, carbon assimilation, carbon reduction, this occurs in biosynthetic phase, also addressed as light independent phase, also known as dark reactions. Calling this as dark reactions is correct term? No, it is a misnomer. Now, drop your pens and listen. Drop your pens, everybody. Listen. First is C3 cycle. C3 cycle is also known as Kelvin cycle, which is named after the scientist who discovered this cycle, that is Melvin Kelvin. How did he discover this? By taking one radioactive isotope of carbon. What is that radioactive isotope? C14 radioactive isotope. He has given this C14 radioactive isotope to chlorella. Chlorella is unicellular green algae. This conducts what photosynthesis? Algal photosynthesis. Tell me, algal photosynthesis is oxygenic or anoxygenic? Oxygenic photosynthesis, because algae are the primitive plants. Oxygenic photosynthesis. When that occurred, first this carbon is fixed into 3-carbon compound, okay, that is 3-phosphoglyceric acid, okay. Carbon is fixed into 3-carbon compound, that is 3-phosphoglyceric acid. This occurs in all the photosynthetic plants. Now you answer me, C3 cycle is discovered by Melvin Kelvin. What is the model organism he has taken? Chlorella. Okay. What is the radioactive isotope he has used? C14. Okay. What is the primary product he has got? Primary intermediate of fixation? 3-carbon compound. That is why it is C3 cycle. First immediate product that is fixed. First stable organic intermediate. When this radioactive isotope is fixed, first organic intermediate is 3-phosphoglyceric acid, which is 3-carbon organic acid. Okay. Clear till here? Now, C4 cycle. C3 cycle occurs in all the plants. C4 cycle is also known as Hutchinson-Slack pathway. Okay. C4 cycle occurs in tropical plants. Where first stable organic intermediate formed is oxaloacetic acid. Oxaloacetic acid. This oxaloacetic acid later turns to malic acid or aspartic acid. Occurs only in C4 plants which grows in tropical climates. Most of them are of monocots. They can be sorghum, maize, sugarcane. Only dicots which are included here are amaranthus. Clear? Most of them are monocots. Only dicots include amaranthus. Clear, right? Next is CAM cycle. Crassulacean acid metabolism. Okay. This occurs, this cycle happens in deserts, where the first stable organic intermediate here is oxaloacetic acid. Then you can have that doubt, ma'am, when first stable product is OAA, why don't you call it as C4 cycle again? Two major reasons. This operates in 2 organelles. We will discuss it in 2 different organelles, but this happens in a single cell. Sorry, this happens in 2 different cells, not organelles, 2 different cells. Mesophyll cell and bundle sheath cells. This occurs in only single cell, that is mesophyll cell. And second major reason is this cycle occurs in most of the Crassulacean family members. Okay, there is a difference in fixation based on daytime and night time. That is the reason this is known as CAM cycle, not C4 cycle. Okay, this occurs in Crassulacean family members. occurs in plants which grows in desert climates. Examples include succulent plants like cactus, agave, opuntia, aloe vera. And tropical epiphytes include orchids. Take down. Yeah, you can. Whoever are not done, you can copy later. C3 cycle, look at here. First, C3 cycle is operated in 3 major steps. Drop your pens and listen. Everybody listen. C3 cycle is operated in how many steps, ma? 3 steps. First is carboxylation, second is reduction, third is regeneration. Okay. For what is meant by carboxylation? Carbon fixation. Carbon dioxide fixation is carboxylation, which is the most crucial step of Kelvin cycle. There is a statement that is given. What is the most crucial step of Kelvin cycle? What is your answer? Carboxylation. Okay. For carboxylation to occur, what are the prerequisites? Carbon dioxide is required and some molecule should accept this carbon dioxide. No? What is that here? It is accepting RUBP, ribulose bisphosphate, okay, which is a 5-carbon compound. And this reaction is catalyzed by what? Enzyme, that is Rubisco. Key factors about Rubisco that you have to know. Rubisco is world's abundant enzyme. Okay, Rubisco, the name itself says ribulose bisphosphate carboxylase oxygenase. This enzyme will have equal affinity to bind with oxygen and carbon dioxide. Okay, it has equal affinity to bind with oxygen and carbon dioxide. For example, ma, there are 5 molecules of carbon dioxide and 5 molecules of oxygen, equal ratio. Okay, then Rubisco will bind with carbon dioxide only. For example, there are 5 molecules of carbon dioxide and 6 molecules of oxygen. Then Rubisco will bind to oxygen. You have to remember that. Okay. What are the key factors of Rubisco? It's world's abundant enzyme. It has equal affinity to bind with carbon dioxide and oxygen. If oxygen concentration is more than carbon dioxide in the atmosphere, it will bind with oxygen instead of carbon dioxide. Whenever the concentration is more, not equal or less. Okay. Then it forms the primary product, that is 3-carbon compound. Okay. 3-carbon compound, which is 3-phosphoglyceric acid. This is 1 carbon, carbon dioxide. When 1 carbon dioxide is fixed, plus 5 carbon, total how many? 6 carbons. First carbon dioxide fixed is 3-carbon compound, that is 3-phosphoglyceric acid. As it is 3-carbon compound, how many molecules will be fixed, ma? 2 molecules of 3-carbon compound. First step is clear, right? What is the primary carbon dioxide acceptor in C3 cycle? RUBP. Ribulose bisphosphate is the primary carbon dioxide acceptor. Okay, which enzyme is catalyzing the first step of Kelvin cycle? Rubisco. Okay, does Rubisco has higher affinity for carbon dioxide? No, equal affinity for carbon dioxide and oxygen. Clear till here? Then 2 molecules of 3-PGA are formed. Okay, they get reduced to 2 molecules of glyceraldehyde 3-phosphate, which is again 3-carbon compound only. For this reduction step, for single carbon dioxide fixation, in this reduction step, how much amount of ATP and NADPH are utilized? 2 ATP and 2 NADPH. 2 ATP and 2 NADPH are utilized. Okay, and this 2 molecules of glyceraldehyde 3-phosphate, which has how many carbons? 6 carbons on total. Okay, from 6 carbon, 1 carbon will be escaping the cycle for glucose formation. If 1 carbon is escaping, rest how many carbons will be there? 5 carbons. That 5 carbons will be regenerated back in the regeneration step. Okay. Regeneration step is crucial to continue the cycle uninterruptedly. Okay. If there is a statement asked to you that which step is the crucial step of Kelvin cycle, your answer should be carboxylation. Which step is crucial to run the Kelvin cycle uninterruptedly, your answer should be regeneration. For this, how much amount of ATP is required, Nana? 1 ATP for 1 carbon dioxide. Okay. So, in a single cycle for fixation of 1 carbon dioxide, you require 1 RUBP rubisco, which forms 3 PGA, 3 phosphoglyceric acid. In reduction, you require 2 ATP, 2 NADPH. This gets reduced to 2 molecules of glyceraldehyde 3 phosphate. And from this 2 molecules of 3 glyceraldehyde 3 phosphate, total 6 carbons, 1 carbon will be escaping the cycle for the formation of glucose. Rest 5 carbons will be regenerated back. So, on total in a single cycle, 2 plus 1, total 3 ATP are required and 2 NADPH, and 1 carbon is escaping for glucose formation. You know glucose has how many carbons, ma?
6 carbons.
So, for 6 carbon formation, how many cycles have to happen? 6 cycles. How many carbon dioxides have to fix? 6 carbon dioxides. Okay. Now, all of you are clear with Kelvin cycle, no? Yeah. So, for 1 carbon dioxide fixation, 3 ATP and 2 NADPH are required. For 1 glucose formation, for 1 glucose to form, 6 cycles should happen. 6 carbon dioxides are required. 6 into 3, 18 ATP are required. 6 into 2, 12 NADPH are required. Okay. First you take down this about C3 cycle, then we will move to C4. Make it fast, ma. Just 3 steps you have to take, that's all. And then bioenergetics. Once this is done, then let me know, I will show you that bioenergetics slide. how much ATP and NADPH are required to reduce 4 molecules of 3-phosphoglyceric acid. 4 molecules of 3-phosphoglyceric acid. 2 molecules of 3-phosphoglyceric acid ni reduce cheyalante enta ATP kavali beta? To 2 molecules of G3P. 2 ATP and that's it, no? 2 ATP and 2 NADPH. He asked 4 molecules of G3P. How much ATP? 4 ATP. That's it. 4 ATP for NADPH. This question chudandi nanu. Amount of assimilatory power required for one sucrose molecule. One glucose it is 18 ATP, 12 NADPH. Glucose has 6 carbons, sucrose 12. Just double it, 30, 36 ATP, done. This is how you will get questions from bioenergetics, okay ma'am? అర్థం కాలేదా? ఇక్కడ అర్థం కాలేదు. In reduction step, for one carbon dioxide fixation, how many molecules of G3P are fixed? 2 molecules of G3P are fixed. They will get reduced to 2 molecules of phosphoglyceric acid is reduced to glyceraldehyde 3-phosphate. How much ATP and NADPH are required? 2 ATP and 2 NADPH. He is only asking you on that one step. To reduce 4 molecules of G3P, how much ATP and NADPH are required? For 2 it is 2, for 4 it will be 4. Okay. Abandama? See, I told you about Rubisco, right? It has equal affinity for carbon dioxide and oxygen. That is what is given here. If it is binding with oxygen, what are the cases that Rubisco will bind with oxygen? You know this, right? What solubility is more? Which solubilizes easily? Carbon dioxide gets easily solubilized with rise in temperature. So, most of the C3 plants are temperate plants. Okay. If temperature rises, what happens? With the rise in temperature, the carbon dioxide gets solubilized and there will be increase in concentration of oxygen. Listen carefully. Once again, most of the C3 plants are temperate plants. Temperate plants optimum temperature will be around 25 to 35 degree Celsius. Okay, more than this, if temperature is rising, what happens is carbon dioxide gets easily solubilized. Carbon dioxides get solubilized, and when carbon dioxide gets solubilized, there will be increase in the amount of oxygen. Oxygen concentration rises, carbon dioxide concentration reduces. In that case, what happens? Rubisco will bind with oxygen when there is rise in oxygen concentration. Okay, clear? In that case, what happens? When RUBP bind with oxygen, another cycle, that is C2 cycle, will be operated, which is photorespiration. We will learn it at end. Now you remember this till here, we will learn it at end. Here only we will learn. Okay, listen. So in photorespiration, this is addressed as a wasteful process. So when oxygen is binding with RUBP, what happens? You see, oxygen along with RUBP. RUBP is how many carbon? What is the enzyme? Is performing what activity? Oxygenase activity. Okay, and this forms two molecules, one 3-phosphoglyceric acid and one molecule of phosphoglycolate. One molecule of 3-phosphoglyceric acid will not do anything. Okay, to get reduced, what should be there? 2 molecules of 3-phosphoglyceric acid should be there to get reduced. Here only one 3-phosphoglyceric acid cannot run the show easily. So, this one molecule of phosphoglycolate, it has to convert to one molecule of 3-phosphoglyceric acid. For that, it has to travel all across 3 organelles: chloroplast, peroxisome, mitochondria. In that traveling, it requires ATP, it requires NADPH. It will not give out any energy, it will not provide any sugars, it will not provide any oxygen also. Rather, it utilizes oxygen and releases out carbon dioxide. That is the reason this is said as a wasteful process. Occurs only in C3 plants. Okay. So, here you look at it, ma. First phosphoglycolate after dephosphorylation gets converted to glycolate. Glycolate enters into peroxisomes. Here it gets oxygenated and converts to glyoxylate, turns to glycine. Glycine enters into mitochondria. Here in mitochondria itself, from glycine carbon dioxide goes out, ammonia goes out, and then this utilizes NADP plus as well. Okay. Glycine gets converted into serine amino acid inside the mitochondria. Serine enters into peroxisome, gets converted to hydroxypyruvate, okay, utilizes the energy NADH, gets converted to glycerate. Glycerate enters into chloroplast, converts to 3-phosphoglyceric acid by utilizing ATP. Phosphate group from ATP gets adjoined with glycerate, turns to phosphoglyceric acid. Got it? It is utilizing the energy, okay. It is releasing out carbon dioxide. Is there any oxygen production? Is there any sugar production? So, this is known as wasteful process. Okay. Usually అదొక టర్మ్ బాగా గుర్తుంటుంది, బట్ ఇవన్నీ కూడా బాగా గుర్తుపెట్టుకోండి. ఓకే, క్లియర్ రైట్. ఫస్ట్ స్టెప్ షుడ్ బి క్లియర్ ఇన్ యువర్ మైండ్. What are the molecules that are formed, and the entire process is starting by phosphoglycolate to get turned to 3-phosphoglyceric acid. Here what you have to remember the most is which substrate is entering the organelle, which substrate is leaving the organelle, where decarboxylation happened, in which steps energy is being utilized, oxygenation happened in which step, dephosphorylation, phosphorylation occurs in chloroplast. Such a type of questions will be asked from photorespiration. Take down quickly. Last benches also can see this, no? Now you can see. Next, no problem, you can write it later also. C4 cycle, listen carefully. To understand C4 cycle, you have to first understand the Cran's anatomy, okay, the structural differentiation that you observe in C4 cycle. Listen ma, mostly in dicot leaf what happens, you will have two types of mesophyll cells, palisade parenchyma and spongy parenchyma. Most of the C4 plants are monocots, we have learnt. There you will find only spongy parenchyma, palisade will not be there, right. So here in C3 plants if you observe, in C3 plants, adjoining to the vascular bundles, you have what? Bundle sheath cells. These bundle sheath cells are connected to mesophyll cells also. How they are connected? Bundle sheath cells, mesophyll cells, they are connected via plasmodesmatal connections. We have learnt about plasmodesmatal connections, right? Yeah. So there is only single layer of bundle sheath cells in C3 plants, whereas in C4 plants, this entire bundle sheath cells will form like a garland. They form a wreath like this. Okay, these are more than 2 layered sometimes. Surrounding the vascular bundles, the bundle sheath cells are more than 2 to 3 layered. Fine, right? C4 plants. This is known as what? Cran's anatomy. Bundle sheath cells will form like a wreath here, like a garland around the bundle sheath cells, around the vascular bundles. Vascular bundles are of what type? Can anybody tell me? Conjoint and closed type of vascular bundles you will observe in the leaf. Bundle sheath cells, what are present in the bundle sheath cells? Bundle sheath cells consists of a granule chloroplast. A granule chloroplast. There are no intercellular spaces in between the bundle sheath cells. They are abundant in the enzyme rubisco. These are the 3 main points that you have to note with respect to bundle sheath cells. There are no intercellular spaces. They are connected to mesophyll cells via plasmodesmatal connections. They have a granule chloroplast, means grana is absent in the bundle sheath cells. Here you see C4 plants. Cran's anatomy of bundle sheath cells I have told you. Mesophyll cells in C4 plants will have what? Granule chloroplast. Bundle sheath cells will have a granule chloroplast. Now you look at the cycle carefully. Everybody can see, right? First what happens in mesophyll cells? Carbon dioxide is accepted via stomatal openings. Okay, carbon dioxide entered into the cell. Once carbon dioxide entered into the mesophyll cell, this gets reduced to hydrogen carbonate via enzyme carbonic anhydrase. Okay, carbon dioxide gets reduced to hydrogen carbonate by carbonic anhydrase. The primary acceptor of carbon dioxide here is PEP, phosphoenol pyruvate, which is 3-carbon compound. 3-carbon compound. There in C3 cycle, what is the primary carbon dioxide acceptor? RUBP, 5-carbon compound. Okay, C3 cycle it is RUBP. Here it is 3-carbon compound phosphoenol pyruvate. There what is catalyzing carboxylation? Here it is pepcase, phosphoenol pyruvate carboxylase. Okay, it doesn't have any oxygenase activity. Okay, phosphoenol pyruvate carboxylase is the enzyme which is catalyzing this reaction, and this gets fixed into 1 carbon dioxide plus 3-carbon compound. Total how many? 4 carbon. 4-carbon oxaloacetic acid. Okay, this gets isomerized to malic acid or aspartic acid. This gets dehydrogenated by the enzyme malate dehydrogenase and enters into what? Bundle sheath cells. Okay, oxaloacetic acid turns to malic acid or aspartic acid by the enzyme malate dehydrogenase. Malic acid or aspartic acid enters into bundle sheath cells. immediately this 4 carbon compound entering into the bundle sheath cells, this gets decarboxylated. Decarboxylation occurs into 3 carbon pyruvate plus 1 carbon dioxide. This 1 carbon dioxide will run the show of Kelvin cycle inside bundle sheath cells. Okay? 1 carbon dioxide is accepted by RUBP, Rubisco enzyme is there in bundle sheath cells, and this will run the show. Next, pyruvate, which is 3 carbon compound, this gets regenerated back into the mesophyll cells. For this regeneration step, there is an enzyme required, pyruvate phosphate dikinase, and how much ATP is required? 2 ATP. So here, how many times carboxylations occurring? 2 carboxylations, one in the mesophyll cell, one in the bundle sheath cells in Kelvin cycle, right ma? And here if you see, how many amount of ATP are required for single carbon dioxide fixation? 3 ATP are required in Kelvin cycle and 2 ATP are required for regeneration of pyruvate. Total how many? 5 ATP. Okay. How much NADPH? 2 NADPH in Kelvin cycle. So total for 1 carbon dioxide fixation in C4 cycle, 5 ATP and 2 NADPH are required. If in your question, how many extra ATP are required in C4 cycle, your answer should be 2 ATP. 2 ATP in extra. Understood everyone? How many needs a repetition? How many needs a repetition? Repetition of C4 cycle. Everykaina malli cheppana C4 cycle? No. Okay kada. Bioenergetics are also clear, right? How many carboxylations?
2 carboxylations.
How many carboxylations? 2. How many ATP are required? 5 ATP. How many extra ATP?
2 ATP.
How many NADPH?
2 ATP.
What type of chloroplasts are present in bundle sheath cells? Agranule chloroplast. How bundle sheath cells are connected to mesophyll cells? Okay, what is the primary organic intermediate that is fixed in C4 cycle? Oxaloacetic acid. That gets isomerized to malic acid or aspartic acid, which is 4 carbon compound. What is the primary carbon dioxide acceptor in C4 cycle? Phosphoenol pyruvate. What is the enzyme that is catalyzing carboxylation? Now energetics. అవదు.
మరి అదే సైకిల్లో అంటే కార్బన్స్ స్ప్లిట్ అయ్యి C3కి వెళ్తాయి కదా మేడం. C3లో అయినప్పుడు ఆటోమేటిక్గా C4లో కూడా అవ్వాలి కదా. కెల్విన్ సైకిల్లోకి వెళ్తాయి కదా మేడం కార్బన్ డయాక్సైడ్.
Listen. Good question, but different గా fragment చేసావు, but a good question. See, bundle sheath cells are present deep inside. Bundle sheath cells are present deep inside the mesophyll cells, and I already told you that is the reason bundle sheath cells have no intercellular spaces, so they are connected only to the mesophyll cells. Bundle sheath cells are deep inside, so mesophyll cell nundi daniki vachedi enti only carbon dioxide. Is there any oxygen present there? 0%, very low percent of oxygen is present. So is there any possibility of Rubisco performing oxygenase activity? No. 100% it will perform carboxylation activity only. Understood now? That is the reason C2 cycle is not operated in C4 plants. Clear? Good. Now you see the energetics. 3 ATP of Kelvin cycle plus 2 ATP of C4 cycle in the regeneration. Total 5 ATP for 1 carbon dioxide fixation in C4 cycle plus 2 NADPH. For formation of 1 glucose molecules, how many cycles need to be done? 6 cycles, 6 carbon dioxide molecules are required. Okay, 5 into 6. How many total? 30 ATP. 5 ATP into 6 cycles, right? Total 30 ATP. 2 into 6. How many? 12 NADPH. Clear? Any doubts anywhere? Clear, right ma? And then here you look into the questionnaire, which is very interesting. How much extra ATP is required to fix 6 carbon dioxide molecules. When extra ATP is clearly mentioned, you have to consider only that 2 extra ATP in each cycle. How much extra ATP is required for the formation of 1 sucrose molecule? 12 into 2, 24. If extra is not asking total ATP, then you have to take 30 for 1 glucose. 2 glucose molecules or 1 sucrose, so it is 60 ATP. Understood, ma? Everybody, take down C4 cycle. Now drop your pens, everybody. Listen, आराम से listen. CAM cycle, crassulacean acid metabolism. This is a photosynthetic adaptation in succulent plants. C4 plants has an adaptation, okay, as they live in high temperature regions, tropical plants, right? Okay, they live in high temperature regions, so they got a morphological adaptation showing Kranz anatomy by bundle sheath cells. These plants are also in desert conditions, right? They also got an adaptation, photosynthetic adaptation of succulent plants. That is, they have scotoactive stomata. Usually in all the plants, C3 plants or C4 plants, they will be having photoactive stomata. Stomata which opens during daytime and closes during night time. Scotoactive stomata opens during night time, closes during daytime. Okay, to escape the heat, to escape the evaporation of water molecules as well. This is scotoactive stomata. Stomata opens during night and closes during daytime. First step, that is carboxylation, that is fixation of carbon dioxide, occurs during night time itself. Okay, carbon dioxide stomatal opening happens during night time. Carbon dioxide enters, gets reduced by carbonic anhydrase to hydrogen carbonate. Carbon dioxide is accepted by 3 carbon compound. What is it? PEP, catalyzed by pepcase, forms 4 carbon oxaloacetic acid, turns to malic acid or aspartic acid. It gets stored in vacuole. What is the activity that happening in the night? Happens in the night? Stomata opens, carbon dioxide fixation happens, OAA, oxaloacetic formation, and they get stored inside the vacuole. This is the activity that is happening during night time. During daytime, what happens is, from vacuole they will again get back to the mesophyll cell. This 4 carbon compound will get decarboxylated to 3 carbon phosphoenol pyruvate and 1 carbon dioxide. This 1 carbon dioxide will complete the Kelvin cycle during daytime, and this 3 carbon phosphoenol pyruvate gets fixed to starch, or they get regenerated back to fix the carbon dioxide during night time. 2 possibilities are there, either they will fix into starch or they will get regenerated to continue the carboxylation during night time. So here in a single cell, 2 cycles are happening, C4 cycle during night time and C3 cycle during daytime. Okay. Okay. So here C4 cycle during night time and C3 cycle during daytime. Clear, right? Any doubts anywhere, ma? Yeah. So this is about SCAM cycle. Now this is the table which is given in your NCERT. Whoever has, you can open it. I don't want to ignore CAM cycles. I have drawn one more column here. First, cell type in which Kelvin cycle takes place. Here in C3 plants, Kelvin cycle occurs in Mesophyll cells, no? Team B, C4 cycle, where Kelvin cycle takes place? Bundle sheath cells. In CAM cycle, in CAM plants, where Kelvin cycle takes place? I am asking you about the cell. See, cell type in which Kelvin cycle takes place. C3 plants, mesophyll cells. C4 plants, CAM plants. Now the question to this team. Cell type in which initial carboxylation reactions occur. In C3 plants. Yes, mesophyll cells. In C4 plants. Initial carboxylation in mesophyll only, no? Here, initial carboxylation in which cell? Mesophyll cell only, in CAM plants also. Simple questions, ma, don't get confused. Third question. How many cell types does leaf have that fix carbon dioxide? Cell types? Huh? How many cell types does the leaf have that fix carbon dioxide? Cell types, only one cell type, because carboxylation happens in mesophyll cells only. Here, 2. Here, 1. Good. Now team A. Which is the primary carbon dioxide acceptor among? Team B. Team C. Which is the primary carbon dioxide acceptor in CAM cycle? You said RUBP, then phosphoenol pyruvate, and you are saying phosphoglyceric acid. Next question. Think and answer, huh? Team A. Number of carbons in primary carbon dioxide acceptor. Here? In primary carbon dioxide acceptor, how many carbons are there? Here? Give chance for Pawan also from team C. This question. Which is the primary carbon dioxide fixation product? Very good. Here? Good. Number of carbons in primary carbon fixation product. Number of carbons. Here? 4 carbons. Done. Next question to team A. Does the plant have rubisco in C3 plants? Yes. C4 plants? Yes. CAM plants? No. Yes. Without rubisco, how is it running Kelvin cycle? Done. Next here. Does the plant have rubisco? Is done. Does the plant have pepcase? No. Yes. Okay. Which cells in the plant have rubisco? Here, which cells in C3 plants? Here? Okay. Next. Carbon dioxide fixation rate under high light conditions. Under high light conditions, does carbon dioxide get fixed? Very low in C3 because they are temperate, no? Rubisco will bind with oxygen. In C4 it will happen as usual. In CAM also it will happen as usual because both of them have adaptations. Next. Whether photorespiration is present at low light intensities? No. At low light intensities, photorespiration is negligible. Next question, ma. Whether photorespiration is present at high light intensities? Yes. In C3, no. In C4, no. In CAM. Whether photorespiration could be present at low carbon dioxide concentrations. Yes. Here? Here? Whether photorespiration would be present at high carbon dioxide concentration. At high CO2 concentration, how Rubisco will bind? No. Here?
Yes.
There is no relation with carbon dioxide, no, in C4 plants. Next, temperature optimum in C3 plants is 25 to 35 degrees Celsius. Here, C4, 40 to above 40, 30 to 40 you can say, 30 to 40. Here above 40. CAM plants above 40. Examples of C3 plants you can take. Most of the dicot plants, all the plants which live in temperate regions. C4, most of the monocots. Maize, sugarcane, sorghum. CAM plants, opuntia, agave, cactus. Clear with the table? About factors affecting photosynthesis, very small concept. Tomorrow I will tell you and I will start you the respiration in plants. Clear till here, no?